Hello and welcome to PH Biology Required Practicals, the ones examiners love because they're testable, marked to a strict scheme, and lose students easy marks when the method isn't watertight. In this video, we're going through all 10 practicals and key tests you need to know for GCSE higher biology. Every method, the variables to control, the expected result, and the traps to avoid. By the end, you'll have the exact wording examiners want. First up, microscopy. Always start on the lowest power objective. Total magnification equals eyepiece times objective, usually 10 times something. If you're asked to calculate the actual size of a cell, use magnification equals image size divided by actual size and rearrange. But watch your units, millimeters and micrometers. And a good scientific drawing has sharp continuous lines, no shading, straight labels, and always a scale bar. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Osmosis in potato. Cut cylinders of equal size, weigh them, place each in a different sucrose concentration, leave for a set time, pat dry, and reweigh. The independent variable, the sucrose concentration. The dependent variable, percentage change in mass. In dilute solutions, water enters, mass rises. In concentrated solutions, water leaves, mass falls. Percentage change is what you plot because it makes different starting masses comparable. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Food tests. 4 to memorize. Benedict's for reducing sugars. Heat in a water bath. Blue turns brick red. Iodine for starch. No heating. Yellow-brown turns blue-black. Burette for protein. No heating. Blue turns purple. An emulsion for lipids. Ethanol first, then water. Cloudy white if positive. Always use a positive and a negative control, otherwise you can't trust the result. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Enzymes and pH. Amylase breaks down starch. Mix amylase, starch and a buffer at chosen pH in a tube. Every 10 seconds, drop a sample onto iodine on a spotting tile. When the iodine no longer turns blue-black, the starch is gone, and record the time. Repeat at different pH values. Optimum for amylase is around pH 7. Rate equals 1 over time, and control temperature with a water bath. This is the classic examiner trap. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Photosynthesis and light intensity. Pond weed in a beaker of water with sodium hydrogen carbonate to keep carbon dioxide constant. Lamp at different distances. Count the bubbles per minute. The rate rises as light intensity rises, but light intensity is proportional to 1 over distance squared. So doubling the distance quarters the intensity. Put a beaker of water between the lamp and the pondweed to filter the heat. Otherwise, you're not just measuring light. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Reaction time. The ruler drop test. One person holds a ruler vertically between another person's fingers, drops it without warning, and they catch it. The distance dropped converts to reaction time via a table. Independent variable might be caffeine, tiredness, or distraction. Repeat 5 times, discard the first, and take a mean. Watch out for the practice effect. Reactions improve with repetition, so always warm up first. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Sampling with quadrats and transects. For random sampling, use a random number generator to place quadrats without bias. Count the target species, take the mean per quadrat, then scale up to the whole area. A transect is a tape measure across a habitat, quadrats at set intervals. Transects show how a species changes across a gradient, like from a shaded path edge into the open. More samples means more reliable estimates. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. The decay of milk. Milk plus universal indicator in test tubes at different temperatures. Bacteria break down the milk sugars, produce acids, and the pH drops. Indicator turns from purple to yellow. Time how long the color change takes. Fastest around 30 to 40 degrees. The enzyme optimum for the bacteria. Cold slows it. Very hot denatures the enzymes. Same reason we refrigerate food. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Culturing microorganisms, a higher only practical. Prepare a bacterial lawn on nutrient agar using aseptic technique. Place discs soaked in different antiseptics on the agar. Incubate at 25 degrees, not 37, to avoid growing anything harmful. Measure the clear inhibition zone around each disc. The larger the zone, the more effective the antiseptic. Use pi r squared for the area if you're asked. Always include a water disc as a control. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. Testing leaves for starch, the proof that photosynthesis happened. Destarch the plant first by leaving it in the dark for 24 hours. Give the plant a test condition, then remove a leaf. Dip it in boiling water to kill it. Heat in ethanol in a water bath, never over an open flame, to remove the chlorophyll. Rinse, then add iodine. Blue-black means starch is present. It's also worth locking in. A glowing splint relights for oxygen, and lime water turns cloudy for carbon dioxide, and a squeaky pop for hydrogen. Have a read through the structure. I'll pause here for a few seconds and then we'll move on. That's every required practical and key test you need for GCSE higher biology. The pattern's always the same. Know the method, know the variables, know the expected result, and know the common error to avoid. If you want to lock this in with 50 exam-style questions and fully worked answers, grab the matching PH Biology Required Practicals Mastery Workbook, plus the full range of mastery workbooks across maths, physics, biology, chemistry at phmaths.com. Simple explanations, serious results. See you next video.